So I will talk about one aspect of our research, and with some, hopefully I get there to something that turned out to be a bit controversial, and then also touch on very recent developments, which I just included because of discussions this morning. So let me briefly set the stage. There is a leitmotif in some of these simulation studies that I see, and these are two sentences from a paper from Paul Dirac in 1929, where he says that the equations to do chemistry and most of physics are known. We cannot really solve them for realistic systems in closed form. And so it becomes desirable that one develops approximate and practical methods to look into complex atomic systems, he writes, without too much computation. Today, we would just say complex systems. And these two sentences somehow summarize the two Nobel Prizes in chemistry, in theoretical chemistry. The first one for DFT, electronic structure methods, and the second one, as it was called, multi-scale methods. Much of the complexity really comes from solving a complicated differential equation, as you see it here in Schrödinger's equation, which over the past 10, 15 years has been replaced by statistical modeling. And so Vladimir Vapnik is one of the important people in this regard, because he wrote a very thick book on statistical learning that foreshadowed these type of developments, learning from data, and eventually turned into machine learning methods as we call them today. And as we will see, these methods are actually rather old, so they go back to the 50s, and people have been thinking about such methodologies for quite some time. So what I'd like today is to focus on a couple of example systems. What the group does is some methodological research on the left-hand side. Multipolar electrostatics is something we have been doing in the past. This has been a bit replaced by more distributed charge models, I call them, but we still use them. And I will show one or two examples. And then most of the time at the moment, we work on reactive force fields of different kinds of flavors, based often on machine learning, but not entirely. Sometimes we also use more conventional empirical force fields to do things more rapidly, as you will see. One of the aims is to put this into software, and this is often included in Charm, or nowadays also PyCharm. There's a Python version of Charm. And then we apply to a range of observational regimes and examples. So I will talk about mainly computational spectroscopy today and some protein dynamics. Only a little about solution phase, reaction dynamics, one example. And so just to set the stage, if we talk about empirical energy functions, we really mean energy functions that idealize the intermolecular interactions in bonded terms and non-bonded terms. And it turns out that keeping a couple of those terms can be advantageous, and then replacing selectively for what one wants to do. Say reactions, one would need to replace a harmonic bond by a Morse oscillator or something that can break. And in the non-bonded terms, as I had already mentioned, our usual conventional energy functions are idealized, we think, in terms of point charges. And if you take a simple example, just for illustration, carbon monoxide. For carbon monoxide, such an energy function misses the entire physics, because CO has no charge and no dipole, and this leaves you with two solutions. The two charges are zero. But if you look at the field around the CO molecule, this is how it looks like from electronic structure. Experimentally, it's known that it has a quadrupole, it has an octopole. These are measured. And so we need to go beyond these simple point charge models. And there are ways to do that. And this has been implemented also by Tristan when he was working in our group in Basel over a number of years and tested. And I will show you one or two examples. So with that, I'd like to move to the contents. So I will show you a couple of examples from spectroscopy and reactions, mainly with myoglobin. And then this topic on hemoglobin, which was a bit more controversial, and then end with one or two slides on outlook. So the first model system that we have been looking recently into, so this was extended over a couple of years also because group members moved, was this trialanine, which is a suitable guinea pig for some of these electrostatic methods, because experiments have been done, for example, in the groups of Tokmakov, but also Peter Hamm has investigated this with 1D, 2D infrared spectroscopy. And so we really can test a couple of scenarios. And the interesting units here are labeled 1, 2, and 3, which are the carboxyl groups that can talk to one another. Chemically different is, of course, number 3 from 1 and 2. And so we designed multipolar force fields, which means that we assign charge, local dipole, local quadrupole on each of the heavy atoms from electronic structure and run simulations. And what comes out of these simulations are frequency distributions, or in particular 1D infrared line shapes. And the arrows on top here, these are the experimental positions of the spectroscopic response. And on the left-hand side, you see the results from using a multipolar parametrization of the force field. On the right-hand side, these histograms come from a DFTB, from a semi-empirical DFTB simulation, and they totally miss the position of these infrared bands. Whereas with the multipolar force fields, we can correctly rank them from lowest to highest. And the most important aspect is the splitting for the two oscillators, 1 and 2, which come about electrostatic interactions on the molecule. And this splitting is 13 wave numbers from simulations, 25 from experiments, so it's not quite there, but keep in mind the parametrization of 1 and 2 is identical, and still we have a splitting. So there is a physical interaction that leads to this splitting. And we can, of course, turn off this coupling by simply calculating the stretch frequencies isolated just from a local normal mode picture, or we can do a normal mode calculation on each MD snapshot on the entire peptide. That's doable because we have an empirical force field, and from that we can estimate the coupling strength between these two oscillators. And that's about 6 wave numbers from the simulations compared with 5 wave numbers from experiments. So the strength of this interaction is qualitatively correctly captured by such simulations using advanced force fields. Now, the advantage we have in a simulation is that we can now also map out other things that you cannot observe experimentally, such as these Ramachandran maps, which you also look at in Frauke's group. And you see there are profound differences between a conventional point charge, think of this as amber, CGN-FF force field, and the multipolar force field. The middle here is this semi-empirical DFT. In the following, I will only focus on the multipolar force field. And this is a beta sheet here. This is more of an alpha helical structure, and you see that they differ drastically between the gas phase and water for point charges, but not so much with the multipolar force field. And the question is, how good is this? And Andrei Tokmakov has been working on this in a very clever way, I think. So he mapped out the Ramachandran map, or the potential, this is the Ramachandran map, using a conventional force field. And then he said, I measured the infrared response, which is the black line. Let me calculate this infrared line shape from an original, so the original energy function, which gives this red dashed line. And what he found is that the peak positions are a bit off and the relative heights are a bit off. And then he did something very interesting. He used Bayesian statistics to refine his map to compute the underlying infrared spectrum. So he used the infrared spectrum as a constraint on his Ramachandran map, and this is what he came out with. And this is rather comparable to what we find here from the multipolar simulations. And he could even put this into numbers by simply integrating. So you can integrate these densities here to come up with percentages the system is in one or the other state. And he predominantly found with a refined force field about, say, 90% in this P2 beta sheet conformation, about 1 or 2% in the alpha, and 14% in the beta. Now, it always depends a little bit how you dissect these distributions and how you set the integration limits. We have a dominant contribution at 98% for P2. We agree that alpha is very low populated. The beta is probably a bit low in our case. And you see, this is really a type of system that has been broadly studied, and all kinds of answers have come up in the past. This is probably a bit more settled now that the dominant population is a P2 conformation. So this is from for a simple model system in these simulations. And this is the current status that our group is working on. So I show you a trained, fully trained neural network potential energy. This is alanine, lysine, alanine tripeptide. We can do MP2 calculations on the full set, train a model, and this is the quality of this force field, of this energy function. You see over 300 kilocalories. This is a very, very good energy function, reproducing. Well, this is the performance of the model. This is the reference calculations. These are the Ramachandran maps. We can run extensive MD simulations. This is MP2 quality dynamics. And you could never do this with ab initio ND, in no way. And what the student then did is put it into solvent. This is gas phase. The energy function is optimized to gas phase. Now we put solvent around it, sample again, and then she calculated for structures, reference data, and compared with the neural network, and you see the performance is still quite good. So this is not fitted to structures that are visited in solvent, and still the force field is quite robust. So this is the current status. This is all not published, but I wanted to give you an idea of where things can go. So now we can routinely come up with MP2 quality machine learned models for atomistic simulations. That's the status we have here. The next thing I wanted to touch, because there is some interest in proteins and spectroscopy and ligands, is some work we have been doing on using spectroscopy to relate thermodynamics with infrared response. The system we chose was lysozyme, and the ligand we used was benzocyanobenzene. There is a reference structure in the PDB for a mutant with benzene, and so we simply replaced the benzene by cyanobenzene. And then we ran simulations, and the background is this paper here in Science from Steve Boxer, where he showed more in a pictorial way what he thinks can be done or what one should find. This here is the measured spectrum of the cyano group, CN, you see it here, in wild type and in the in a mutated protein. Everything in color is the electrostatic potential in the wild type and in the protein. And what he says is, depending on whether you are in the wild type, black, or in the protein, you expect spectroscopic shifts of the order of a few wave numbers. Here, the delta is about 4 wave numbers. So we are able to compute such spectroscopic response to this accuracy nowadays. And what it basically tells you is that you can relate structure with spectroscopy. And the question is whether you can also relate it to thermodynamics. So what would be nice is, of course, what you usually assume is the stronger the field, the stronger the perturbation on your spectroscopic signature, and the stronger the effect on the binding strength. And it would be attractive to replace a complicated experiment such as ligand binding by a simple 1D spectroscopy experiment. It would be fascinating. And so we went through this routine and determined the frequency shift between wild type and 2 mutants. And you see that these frequency shifts are really of the order of a few wave numbers, as predicted by experiment. But at the same time, from simulations, we can also compute, of course, the change in binding strength, and you see that the change in stabilization relates to the change in the frequency shift. So there is a correlation between frequency shift and interaction between the protein and the ligand. We also did 2D infrared spectroscopy, but we were unable to find any signatures. So there are a couple of signatures, such as the decay times of the frequency correlation functions, but we didn't see anything particularly compelling. What, on the other hand, we were able to compare directly with experiments is this static component here. So this is the level at which the frequency correlation function decays, doesn't have always to decay to zero. We will see this in a minute. And this static component, two wave numbers, converted from picosecond to the -2, is consistent with every other static component that was measured experimentally in Fayer's group in Stanford for different types of compounds. So these are all very realistic characteristics of these frequency correlation functions. And because we saw this, we thought we want to look at the more general problem of using labels, small labels, to characterize local dynamics in proteins. Again, lysozyme, and we decided to use azide, so here the N3 unit, to label all the alanines and see whether the dynamics, the spectroscopy is different depending on where you put this label. And that would be a first step to label local dynamics from a spectroscopic measurement. Now, to do this, we need accurate potential energy functions for the label. The label is a triatomic molecule, and one of the ways to construct such very accurate representations are so-called kernel-based methods. Now, these kernel-based methods, they were reintroduced in physical chemistry by Herschel Rabbits, but they actually go back much longer. So in 1950s, Aronshine wrote a very memorable and important paper on this topic. It's cited about 8,500 times now. And this really launched the whole business of kernel interpolation, as it is called today. The idea is relatively simple. So think of the following problem. We have an arbitrary function, which is a black line that you cannot see. And then we take samples from this black line. This is what you usually do when you compute, say, a potential energy surface. You take structures, you do a reference calculation. This is one of the circles here. And then you have an engine, a machinery that reconstructs the entire curve from discrete data. And that's called kernel interpolation. And what you need is a so-called kernel matrix, which is a recipe of saying how much does a certain point know about the value of the function at another point. And this is modeled in Rabbits' language as a so-called confluent hypergeometric function here, this F12 function, which are algebraic functions. And we can construct this kernel matrix, which has dimension n by n, where n is the number of points. And then we have sample energies, which are our computed energies here. We need to invert this matrix to obtain these expansion coefficients. And once we have them, we can reconstruct the entire curve. And this is the red line, which superimposes the black line. This is why we don't see it. So what we learn from this is that it is possible to reconstruct very accurately a global function from discrete data. And this is very powerful. And that's what we often use for small systems. There is also a toolkit on the internet, which can be used for problems up to about 10 to the 6 energy points. And this is the performance for azide. So you see over 120 kilocalories, you simply have a straight line. The correlation coefficient here is 0.99. So this is very, very accurate. This is as accurate as a reference method, which in this case is a multi-reference CI calculation. And then we compute spectroscopic observables. And what we find is, for example, if we look at the table, the experimental frequency of the asymmetric stretch is 2457, and the calculation using solving this 3D Schrödinger equation is exactly the same number, 0.2 wave numbers away. What is a bit different is the width of the line. This is always a problem from spectroscopy alone compared with experiments, but it's reasonable. So this is a relatively good comparison. And we get, for example, the correct change in the lifetimes on the picosecond timescale in water and D2. So with this, we can work. What I show you here is how the gas phase potential surface looks like. This is, of course, idealization. Now we put this label onto the protein, and it's not alone anymore. So we recomputed the entire potential surface with an alanine linked to the N3. And you see that the global shape of the potential surface is relatively well reproduced. One could now, if one wanted, also parameterize this. We didn't do that. And then we ran MD, calculated spectroscopy, and what you see is that every label here, 9 to 111, these are the alanines. Each label has a different absorption spectrum, which means that this is specific for a particular site on the protein. And of course, what the experiment measures is some sort, if you don't selectively label, is some sort of a average of that. Now, is this realistic? For this, I cannot compare with experiment. We had done other work on insulin, which shows the following picture. There we did the full story. So this is all 102 CO labels of the amide 1 unit. And each of those, the colored lines, are individual CO positions, and the black line, this is what the experiment on insulin is supposed to measure, because unless you label, and so this was a labeling experiment here by my colleague Mike Weiss, he labeled one of the COs with C13, which is an awful lot of work to do a specific carbon label. And the question is now, how realistic is such an overall line shape? And this has been measured, that's shown here, that's the black line. Black line is solid here. And all the other lines are computed with different methods. The one that is most relevant is the normal mode, the red line. And you see, again, our line width is a bit too narrow, but at a factor of about 50%. But it is absolutely clear that this black line here that is measured consists of a superposition of many different local CO signatures. Now back to this lysozyme, for which we don't have real experiments. What I show you on the right-hand side are these frequency correlation functions, and they show really some of them decay nicely to zero, but others have this static finite time or infinite time saturation, which means that there is some dynamics that does not decay on the 5-10 picosecond time scale, which means that the system is frustrated in these positions. And so this is the status of such position-resolved spectroscopy. We have now also looked at the coupling to the low frequency mode, and we found some correlation between slow breathing motions, as for example, Heidelberg looks at in Frankfurt, to the IR measurements for exactly these type of systems, and there we can potentially compare with experiments. So this was spectroscopy. Let me move on to reactions. There are different ways of doing things. What many of you will know is ab initio MD, which often is done at DFT level. There are a couple of technical aspects, such as basis set superposition error, converging the Hartree-Fock, choosing an active space if you do it electronically excited states. And then there have been alternatives that have been developed over the past, for example, EDB methods. Javier Varshel has done this, ReactFF, some of you may know. We have our own flavor, which I'll briefly describe in the next slides. And then we started to work with kernel-based methods, in particular NN-based methods, but mostly for gas-phase systems. So this is our reactive ND implementation. What we think of is that we start with one connectivity in a molecule, and then we break a bond, and we have a new connectivity. So these are two independent force fields. For example, this magenta line and this black line. They are two different energy manifolds. We can also have a third state, like this blue line. And what we then do is write the total, the global potential, as a weighted average of all these states. Now, whenever the difference in energy between one of the states and all the others is large, you should only consider this state with the highest weight. It's like a Boltzmann sum. Clearly, for this structure here, the black line is the lowest in energy, the blue is higher, and the magenta is off the charts. And so the weight will be 0 for magenta, it will be very small for V1, it will be almost 100% on black. As you approach a crossing, the surfaces that are closer in energy start to contribute 50-50. And that's seen in these weights here. So this surface 2 changes from weight 1 to 0. The other two surfaces from 0 to something finite and eventually 1. And if we do a weighted average, we get the red line. The weights are analytical functions of the coordinates. We can compute forces, derivatives. And so we have a way of running energy conserving, reactive MD simulations. And that's what we will do in the following. So this is done for two sample systems. One is hemoglobin with nitric oxide, which is relevant physiologically for vasodilation, for example. And the process we looked at is the rebinding of nitric oxide to myoglobin. Now, many things have been done for this system, but not everything. So, for example, one observes two rebinding timescales, and the question was, what are they due to in conformation space? These are the two surfaces of the ground state and of the electronically excited state. We need both of them. We use reproducing kernels, just what I explained before, in 3 dimensions, for NO interacting with the iron. So this is, again, the formula. And then we run ND simulations on that in full solvation. And what we observe is, let's look at the right-hand chart first. The green dots, the green trace is most relevant. This gives the correct protonation state of the histidine residue. So this is this histidine coming from top. It's not drawn here, we will see it later on, but this is histidine 64, which is known to be in an epsilon protonation state. And what we see is that the fraction of rebound NO molecules as a function of time increases, of course. And this cross here is an experimentally determined fraction, which agrees rather nicely with the simulations. On the left-hand side, you see an interesting comparison. So this red line is from simulations. It measures the amount of iron relaxation once the NO leaves. So once the NO is away from the protein here, the iron needs to relax because it's 5-fold coordinated and not 6-fold. Now, this relaxation process has been measured experimentally. So these are the time constants, 3.5 picoseconds, which agrees with consistent with the simulations. And the longer time scale, so there are 2 time scales, is 65 from simulations and 85 from experiments. It's rather nicely consistent. When we do this now explicitly with experiments, all the colored traces are from experiments. The upper traces in water, the lower traces in heavy water. And you see that the simulations, which are the open symbols, are really nicely consistent across as a function of time. So we can follow the kinetics of ligand rebinding from a microscopic model in reactions. And to even more support these simulations, we also computed the so-called X-ray absorption spectrum, different spectrum for the ground and excited state. The symbols here are from experiments, and the solid lines are from the calculations. So we take samples and compute the X-ray absorption spectrum with software, and this is really nicely consistent, which means that we can now go into analysis and interpretation. What are the two timescales? The 10 picosecond and the 100 picosecond timescale. And if we just focus on the two upper panels here, the top trace here shows you the localization of the nitric oxide ligand relative to the heme group. Iron is the reference atom. And the lower trace gives you the positioning of this histidine 64, which is now this residue here, this histidine epsilon. And what we see is that for rebinding trajectories on short time scales, the histidine hangs out to the left upper corner. Whereas for longer time scales, it starts to sample this state called B, which is also a so-called spectroscopic substate, has been characterized in the 80s. And so really the difference between a short and a long rebinding trajectory is the positioning of the histidine at the moment of excitation. So we can correlate structure with spectroscopy. And that's the basic message of this piece of work. We then decided to go one step further, do something slightly more complicated. I only have one slide on this. This is really the interaction of another globin. In this case, it is truncated hemoglobin, and they are all somehow related in vasodilation. And the process of interest is ligand exchange, changing from NO plus O2 going to O2 bound and NO3 ligand exchange. We use exactly the same procedure, reactive MD, and compute something like lifetimes. So this is the free energy profile from umbrella sampling. It is known that the overall process is slow, about 1 per second, and that goes from right to left. And you see the barrier is 20 kilocalories, and 20 kilocalories is 1 per second in rate. On the other hand, the time constants computed from MD simulations is 4.5 times 10 to the minus 3, which falls in a range of observed rate constants. And we can also compare the differential change in free energy between the tyrosine and alanine mutant at a particular position, which agrees rather favorably with experiments. So this is another process we're able to look at explicitly with MD simulations. So with that, I'd like to come to something a bit larger in terms of reaction. So this is the Claisen rearrangement, where we were interested in how this reaction, so you see Claisen really is done with chorismate, and then we start to amputate, not only we, also people in the field, we amputated, took away CO2, or added the CO2 instead of the phenol group, and then we further amputated to allyl ether, and studied this in different solutions. So here you see how these empirical mixed energy functions perform. Of course, they have not a one-to-one correlation as these machine learned energy functions, but for example, the minimum energy path here is extremely well modeled. So the black line, the black dots are electronic structure calculations. The red line is the minimum energy path from the reactive phosphate. And we can now study this as a conventional MD simulation in gas phase, in water, in the protein. These are the different colors, and see the barrier reduction as the polarity of your solvent increases. And we can even put this chorismate into its native protein and check what the barrier height is. So this is from simulations, the red line. The experiment is a bit higher. So it's still a force field. And so one probably needs to readjust it a little bit. But what I found quite interesting is to see how the delta G in water and chorismate start to change. So here you see the catalytic effect of chorismate as the protein, because for the two other substrates, the delta G is unfavorable, whereas for chorismate is actually favorable, and which means that the protein does its job. And the explanation for this here is that entropy is extremely large in these two systems. If we put this into Chorus rate, there's a lot of free space, and the transition state cannot be stabilized as it can be for the native substrate. So this brings me to the last topic. This is hemoglobin. And this was really spurred by something that Martin had asked a long time ago. He said, look, I'm confused. We have all these force fields, but some of the most important things for me, it doesn't do right, and I don't understand it. It has to do with the stability of two conformational substates of hemoglobin. So T is the terse state, R is the relaxed state. The subscript means the number of bound ligands, which is 0 in this case, so no oxygen or CO bound. And there is literature, very old literature from Stuart Edelstein, who wrote that one can compute this thermodynamic stabilization of the T state relative to R. The barrier is not known, but what the transition time is estimated to be about 20 microseconds. And then there are the MD simulations, which consistently say our work shows that the R structure is more stable than T. The surprising result is the instability of the T state, its tendency towards R. So this was just found and stated as a fact. And so Martin's question was, what can we do about it? And we tried all kinds of changes to the force field. Maybe something is wrong with the iron parametrization. And eventually we came across this paper here by David Chandler. Many of you will know about the hydrophobic effect, which charts out the structure of the solvent around a spherical particle as we change the size of the spherical particle. And you see that the solvent structuring changes profoundly between a small and a very large particle. And this led to the idea of looking at the solvent. And what we realized was old work by Gerhard Hummer, who showed, for example, that there are very strong finite size effects on the diffusion coefficient. If you have a small box of water, a large box of larger box of water, the diffusivity just keeps on increasing. And he worked out, and Kurt Kramer actually has a much earlier paper in 1993, going in the same direction, that there are finite size effects proportional to 1 over L. And if you finite size correct all of those according to this formula, these are the resulting diffusion coefficients. And so we decided to use increasingly large boxes of water and look at the stability of the T state. And so this is what comes out. This is the one of the particular distances we looked at, 2 histidines, the green line here. Whenever the distance falls below 30 angstroms, the hemoglobin collapsed towards the R state. So these are simply crystal structures. That's why we use R4. The structural rearrangement between R4 and R0 is known to be small. And what we saw is that as the box size increased, the protein lived longer in this T state. We can plot this as a function of system size. So we looked at the water diffusion coefficient, and you see the water at 75 angstroms is just fluctuating around the corrected value from Gerhard. Whereas in the protein plus water system, in the smallest system, the water is much too slow. And then it starts to pick up speed. And it approaches the finite size value. And if we then plotted the lifetime, as per our simulation, on the x-axis, relative to the diffusion, you see it is almost a straight line. The 150 didn't decay, so we just plotted the last point, which was 800 nanoseconds in the simulations. And this was just an empirical finding, which we found was interesting. It spurred a lot of discussion later on, which I actually liked. I think it is important. This is not to say that it is the only reason to explain why T0 is stabilized in our system. We would need to run many more trajectories, absolutely clear. But at the time, it was simply not possible for us to we don't have the infrastructure to run. At that time, we also did a couple of follow-up studies, multiple microsecond simulation on our clusters, but we found this rather at least stimulating. So with that, let me come to an end. A couple of notes, maybe also related to one of the discussion points this morning. I think in terms of protein dynamics, there is very remarkable work by Alex Tscharchenko, Klaus Müller, and Oliver Unke, who presented some real biomolecular dynamics from machine learned force fields. What I show you here is from the original work from Oliver and me 5 years ago. We did alanine 10 folding already at that time. It was possible to do, to train and run simulations. The technology I have shown today is implemented in CHARM. This paper just came out as a status report, what CHARM is able to do nowadays, and how to interface CHARM with machine learning energy functions. And as a last few notes, just to show you where gas phase simulations are and how the different approaches actually compare. This is now a gas phase reaction. What I just want to illustrate is how this empirical reactive force field performs compared with the machine learned force field. Of course, the spread is much larger. We would need to introduce more parameters to make this better. But again, the minimum energy pathway is nicely reproduced. And if you look at the timings, so running a nanosecond of such a gas phase reaction takes an hour. Using the left-hand side, it takes a day on the same computer using the machine learned force field, and it takes 1,000 years to run it at the level of theory, which was used to parameterize these two energy functions. And if one looks at the observables between these two energy functions, they are basically vanishing. So the differences in the observables is very small. I don't discuss them here. One of the downsides of these types of energy functions is still that they are horrendously over-parameterized. So such a neural network has about 1 million parameters. I mean, this energy function will have about 100 million parameters. Very over-parameters, and therefore it's relatively heavy also to evaluate. And so one of the most recent steps was to do these tiny neural networks, which combine what I just have been talking about, these kernel methods, reproducing kernels, with a small 3-layer neural network. This has about 1,000 parameters. These are the learning curves, which tell you how well the model reproduces the energies and forces as you increase the training set size. And FISNet is a neural network, potential energy, and the two colored lines are these minimalist neural networks. You see the training curves between red and gray. They are very comparable for energies and forces. And the interesting thing is now to compare evaluation times. So if we use FISNet, an energy costs about 34 milliseconds for, in this case, this molecule oxalate. And this decreases to 1.3 if we use Python. And the nice thing about these minimalist neural networks is you can also implement them in high compute language, in Fortran, for example, and then you get another 2 orders of magnitude. So it's really performant. Huh? And this is implemented also in CHARM. And you see the performance. Here are the errors again. They are 1 kilocalorie, less than 1 kilocalorie across the board over many tens of kilocalories. And so this is one of the hopes we have for the future, at least for small molecules, to use such minimalist neural networks to be really efficient, have them implemented not in Python, but in Fortran, which may be more accurate. You hear different things here, but yeah, I like that. And last note on a paper that was just published by Luis last week. I think one of the important aspects in the future is to critically look at these machine learning models to uncertainty quantification. And this paper here discusses uncertainty quantification models, such as deep evidential regression, different flavors, so-called ensemble method, where you train multiple independent networks, and Gaussian mixture models. And these lines here, they tell us how many outliers, how many badly performing structures in a pool of data is found if you are looking for the top 100 badly performing or top 1000 badly performing. And what is the conclusion is of this chart is that such ensemble models, these blue lines, they perform best. We are always recovering, finding most of the culprits in the data set, whereas such deep evidential regression methods in their simplest variant, they are not very reliable. And other methods such as Gaussian mixture are actually also performing reasonably nicely. So, and this is a topic that I think in the future needs more attention, uncertainty quantification. So I hope I could just give you an overview of what we do. This is part of the current group. So many people on this. So Luis is here also. He worked on many of the aspects of uncertainty quantification in my group. Silvan did all the minimalist neural networks. This is an older picture. Let me see. So Debashish was instrumental in these kernel-based methods. Oliver did all the work on the neural networks. Mariam did all the spectroscopic labels, insulin, N3, and Kailan did some of these Claisen rearrangement reactions together with Sebastian. I thank them, I thank you, I thank those who gave the money, and if you have questions, let me know.